Hello students, how are you? Today we are going to start with genetics and variation. In that the first chapter is inheritance and variation. So we are going to start with quick revision with this chapter and we are going to learn some basics of this chapter. And they are very important in NEET, CET and ICER exams. So please understand this. I have made in very smaller and smaller part for you all. Today we are exploring why and how of biological similarities and difference. Inheritance is process by which traits are passed from parents to progeny. It is very basics of heredity. On the flip side, we have variation, the degree of which offsprings differ from their parents. Even siblings are not identical. Today we will find out why. So first topic is Mendel's law. Let's talk about Gregor Mendel. He is the father of genetics. He worked with the garden peas and chose 7 pairs of contrasting traits. Why peas? Because they have short lifespan and clear opposing trait like tall vs dwarf or round vs wrinkled seeds. So he discovered some laws. The first law is law of dominance. In this law, in a heterozygous pair, and one allele and the one dominant one masks the other, which is recessive. This is why an F1 hybrids look like one parent, not both. Okay. So the second law is law of segregation. This is universal. Okay, it states that during gamete formation, the two alleles from a trait separate or segregate. A gamete carries only one allele. There is no blending here. The draft trait hidden in F1 reappears in F2 because the alleles are separated safely. So next law is law of independent assortment. In this, in a dihybrid cross, like it is like looking at two traits at once like color and shape, the inheritance of one trait does not affect the other. They sort independently. This is how we get new combination like round green or wrinkled yellow peas. Post-Mendelian discovery. He discovers two dominants. First is incomplete dominance and codominance. In incomplete dominance, like take an example of snapdragon flowers. They are red crossed with white gives pink. That dominant allele is not strong enough to fully mask the recessive one, resulting in a blend. So now we are going to learn about codominance. Looking at a human ABO blood group, if you have A and B allele, you are blood type AB. Both express themselves fully and simultaneously. So next topic is chromosomal theory and linkage. By the early 90s, scientists were looking for a physical, like home of Mendel's factor. Walter Sutton and Theodor Boveri observed that chromosomes behaved exactly like Mendel's factor during meiosis. They proposed the chromosomal theory of inheritance, stating that genes are located on chromosome and the segregation and independent assortment we see in traits are actually caused by a movement of this chromosome. But if genes are on chromosome, what happens when two genes are on the same chromosome? These were T.H. Morgan and his fruit files come in. Morgan noticed that some traits does not assort independently. He discovered linkage, the physical association of genes on a chromosome. So the first linkage is tight linkage. When genes are very close together, they rarely separate or inherited together as a unit. This keeps the parental combination intact and reduce variation. So the second is recombination. Sometimes linked genes do not separate. This happens during crossing over in meiosis. The further apart two genes are on a chromosome, the higher the chance they will swap places, creating recombinant, means non-parental combination. So the next is gene mapping. The Morgan students, Alfred Surtevant, realized he could use this recombination frequency to map the exact position of genes on a chromosome. It's like almost like a GPS for genetics. So we have finished here one short inheritance and variation chapter. Hope you like all my video. This is the part 1. Soon I will put part 2 of molecular basis of inheritance. Thank you.